introduce our next speaker, who is Michael Weigand from the, I hope I pronounced that correctly, from the CDC, and will also be talking about generating pipelines and sequence assembly and annotation for monkeypox virus assembly and annotation.
You did, in fact, do a great job of pronouncing my name. Thank you. Can you see my slides okay?
That looks good. Thank you.
Okay, great. All right, so you can see here that I currently serve as the lead for our bioinformatics and pipeline development unit within the CDC's monkeypox response. I will out myself up front that I am not a virologist. I am a bacteriologist, and most of my day job is studying the genomes of bacteria. And so I am grateful and humbled to be included on this agenda today. So I'm going to talk to you a little bit about the development efforts that we have had within our response structure towards producing good monkeypox virus genome sequences. So although the CDC response to the current outbreak started sometime in May, it wasn't until recently in July that we really stood up a unit focused on bioinformatic development specifically. And our goal can be summarized here that we're trying to develop an automated and high throughput pipeline for sequence analysis and importantly, public database submission. I think you can trace this really to just the shift in scale of our genomic surveillance. So our pox virus and rabies branch previously was probably sequencing on the order of 10 to 20 genomes a year, and now we're at a scale of something like 100 genomes a week. We just heard that the monkeypox virus genome has a lot of interesting repeat features that can make assembly quite challenging. And we've heard many times already today about these inverted terminal repeats. And here I've just highlighted a couple of others. Complex repeats that we see throughout the genome. And so, sequencing these with a combination of short read and long read technologies and going through a manual curation process makes sense at a scale of 10 or 20 per year, but certainly is not practical in our current climate. So this overview summarizes our pipeline development, which really kind of can split into these four main functional categories, which I'll go over. But importantly, our input here is shotgun metagenomic data, typically from a NovaSeq 6000 instrument. And you can see that I've listed a number of open source software tools here for each of these steps. At least some of these are probably familiar to members of this audience. Our pre-processing takes place as two steps. We do a taxonomic classification using Kraken-2. We found that using a database that comprises both a monkeypox virus and a human genome reference does perform much better than either of those individually. Specifically, it allows Kraken to more accurately filter some reads from these small areas of shared homology. And we can observe that produces cleaner downstream results. We're thinking about ways of making that more generalized to cover a broader orthopox virus reference collection. But so far, we're seeing just good results with monkeypox virus only. And we've gone through a few iterations to test and confirm that this approach also does well to remove sequences from other microbial pathogens that may be present in those samples, like STD agents. After which, then we're just using pretty standard read trimming and filtering using FastP. Those monkeypox virus positive reads then go into parallel reference-based consensus calling and de novo assembly using unicycler. Our plan is to merge these into a single final assembly through a process similar to the manual review that has been conducted by our poxvirus and rabies team previously. And most of our development effort right now is focused on parsing through the assembly graph structures that Unicycler produces. Because we've seen that most of these de novo assemblies are fully connected. So we think we can use this to robustly stitch together single contigs. If you were to open one of these graphs in a visual exploration tool like Bandage, I think it's pretty intuitive to see how you might peel apart these collapsed ITR contigs in the highlighted dot boxes there. And so we're trying to come up with a way to programmatically walk this path through the graph linkages to produce a final contiguous sequence. And then we'll also probably do working on some additional refinement to estimate those repeat copy numbers. You can see from my highlight here that Unicycler often will recover some of these repeats with a normalized coverage relative to the rest of the genome. So we think that also provides a good first approximation of those repeat count numbers. And lastly, I'll just mention that Crystal shared a few examples this morning where we've recently observed structural rearrangements and deletions within some of these outbreak genomes. And so far, it seems that this graph-based approach actually does pretty well at resolving those assemblies accurately. We take those finished assemblies and then annotate them according to a reference using a tool called Liftoff. This approach basically uses a Minimap2 alignment to transfer those annotated features from a close genome. Here we're using that RefSeq sequence that Aneta discussed, and I think she did a great job of emphasizing the importance of using that standard nomenclature and those OPG ortholog names. So all of our annotated sequences will conform to that standard. We find that ITRs can be a little bit tricky to detect accurately by this method, but otherwise we see that just with the default settings of this Liftoff software, we're able to capture pretty much all of the CDS and miscellaneous features. And then lastly, we perform a programmatic submission to NCBI. And this is a modification of the seq sender pipeline that was developed by one of our bioinformaticians here at the agency, Dakota Howard, that really is the foundation of almost all of the SARS-CoV-2 sequence submissions from CDC surveillance activities. And seq sender gives us this ability to perform simultaneous and linked submissions to biosample and GenBank and SRA in a very standardized and high throughput manner. We're not currently submitting directly to GISAID, but as been noted in the chat earlier, all of our GenBank sequences do eventually get pulled into EpiPox. And I've put the bioproject number here if you're curious to see where all those data get organized. And of course, briefly, I think it's always worth mentioning that the way that you construct these pipelines is also important. We're using Nextflow because that provides a scalable and reproducible workflow management system that conforms to many of those bioinformatic best practices that this group is familiar with. Most of the open source modules in our pipeline are being pulled directly from NF Core, which is a community-driven platform and provides a lot of good peer-reviewed pieces that you can build into your pipelines. All of which makes this pipeline hopefully more portable. And we're looking forward to being able to share that eventually. And we know that it's familiar to many public health bioinformaticians, in particular used by some state public health laboratories. And of course, finally, I'll just point that the pipeline allows us to process an entire set of NovaSeq data in about 2 hours, returning the natural order where we see that the sequencing step is actually rate limiting, not the bioinformatics. Of course, thanks to all the many great colleagues across the agency, as I mentioned, I'm currently working on this as a deployed to the response. So it's great to be able to interact with a lot of folks that aren't in my usual pathogen specific orbit. And thanks again, Anna, to having me too.
Thank you, Mike. Well, that was a fantastic talk. And there are a ton of questions in the chat. Since we've got some time, we could try reading those out, or if people would like to unmute and speak up, that is totally fine too.
Hey, Michael, this is Kevin. If you don't mind, Anna, if I ask the question. Yeah, I just want to make sure I get that clarification on Seek Sender. Is that currently being used to submit to GenBank? Because I think it mentioned the FTP, and Dewani was making some clarifications, but maybe we can just have that open conversation here.
Yeah, sure. So Seek Sender will submit to biosample, and it will submit to SRA, and then it will actually produce the submission file for GenBank. And so, it provides that good connectivity because it gets the biosample accession and makes sure that that's all synced correctly before you run table to ASN. But yes, thank you for that clarifying question. We do end up having to email those ASN1 files to GenBank to get them submitted.
Ah, got it. Thank you for clarifying. Really appreciate that. Great presentation, by the way.
Thanks.